which I meet. You have to look what the title is of your lecture. This is for you. Are you using the microphone? Okay. And you are, I'm sorry, yeah, Dr. Herrmann. So you're from Switzerland and from the Swiss Federal Laboratories for Material Science and Technology, and you are going to tell us something about magnetic blood purification. Is that correct?
Yeah, that's right. Thanks so much. Good morning, everyone. Thanks for being here this early. I'd like to discuss with you today a couple of challenges we are facing when trying to translate magnetic blood purification. So just really briefly, I'd like to recapitulate the concept of magnetic blood purification. So the basic idea is that we capture toxins or pathogens by using magnetic beads, and then we can remove the pathogens or disease-causing factors immobilized in the magnetic particles by magnetic separation. We've done quite some work on trying to translate this process and to make it more amenable for clinical translation. So we've implemented the concept into a continuous extracorporeal circuit. So the idea is that in future, blood would be drawn from the patient. It would circulate in the extracorporeal circuit. We would inject magnetic particles. The particles would bind the pathogen. And before the blood is recirculated into the patient, we would remove the magnetic beads. In 2013, we've done that also in vivo in a rat model, and it worked quite well. So we can show that the toxin or pathogen concentration actually decreases in a reasonable amount of time. So what we've learned over these past few years are that there's still a couple of limitations and remaining challenges. Number one is when we want to move to bigger targets and more clinically interesting things like bacteria or cells, we really push the kinetic limits, meaning that we have a very limited time in the extracorporeal circuit, so binding times should be in the order of a couple of minutes. Number two is particle safety. So we need to show, or we need to figure out what happens if particle recovery is not 100%, and especially if we accumulate particles over time. So if we have extensive and repeated treatments, we might accumulate quite a few particles in the body. And number three is, in order to be able to remove a pathogen, we need to functionalize our beads accordingly. So we need to know what pathogen we need to remove. So basically, one of the biggest challenges is the prior identification of the disease-causing pathogen. So these are the three main challenges that might jeopardize the risk-benefit ratio. And we've tried to address these challenges. So the first one, binding times, limitations in binding times when removing bigger targets. We've joined up with Marco Latuada at the Adolf Merkel Institute. So we've joined up with Marco Latuada from the Adolf Merkel Institute, and he has done some modeling, and he's looked at bacteria-particle interactions. So what we are looking at here is bacteria with a certain number of magnetic particles attached to them. So the black line here basically shows the number of bacteria with no particles. And we can see that this line drops to 0 at 0.2 minutes. Then we look at the line where one particle is bound to a bacteria, the blue line. 3 particles bound to a bacteria, the green line, and 6 particles bound to one bacteria. So these are the different populations. And what you can recognize here quite clearly is that there is a window of opportunity where we have reasonable binding within reasonable binding times. Challenge number 2, particle safety. When we think of safe particles, we need to optimize 3 things. So we need to have the particles degradable, we need to have them highly magnetic, and we still want to have them small, so to have good mobility or diffusivity, to have rapid binding. So in this parameter space, there are different options. You can use either metal particles, metal oxides, different sizes, of course. And we've recently started to look into magnetic nanoclusters because we believe that these particles have the advantage that they can rapidly disintegrate and then be rapidly degraded as well. So these are clusters of tiny magnetic metal oxide beads. Challenge number 3, pathogen binding. And this was really something that happened by pure serendipity. So I met Gerald Peer at the conference on staphylococcal diseases. Gerald Peer developed a PNAC antibody, that's a polyacetylglycosamine antibody that binds to virtually all pathogens. It binds to 10 or actually 9 of the 10 most common pathogens causing nosocomial infections, and that allows us to bind most bacteria, actually. So it's a really, really interesting capturing agent. So now we have to combine everything and do the experiments. So we've started with magnetic nanoclusters. We functionalized them with the PNAC antibodies by using protein HE hybrids, actually. And we've looked at the colloidal stability, and we can see that we get fairly good stability even in the presence of serum, so that's good. And then more importantly, we can actually employ these beads to capture bacteria. That's what we see here. So we can see that we reach really high capturing efficiencies, and we can decrease the number of bacteria quite a bit. And more importantly, we can also show what bacteria we have captured. So in that case, we can show that it's actually Staph aureus, and we can do PCR analysis and identify the bacterial strain. Then thinking of a continuous process, we need to show that we can also recover the beads at a very high separation efficiency. And we've looked at different separation times. So ideally, separation time would be minutes or shorter. And we can see that we require about 5 minutes to capture the beads. 1 minute is not enough, so there is some room for improvement here. And similarly important is, of course, the compatibility of the beads with cells. And we've looked at the viability of monocytes. And we can see no change in viability, which is good. Then next is particle uptake. So we looked at particle uptake as a function of time, also in macrophages. And we can see that within 10 minutes, which would be a very typical contact time of beads with monocytes or macrophages, we have very little uptake. But if there is longer contact time, of course, the beads are taken up, which is also visualized here. So we can see significant uptake at 24 hours. And then as a last thing, of course, it's very important to show that our beads actually degrade with time. So we did a degradation study according to the method initially described by Torres Scotland. And we can see that the beads degrade within reasonable time frames. So this brings me to the conclusion that the PNAC antibody functionalized beads allow efficient capturing and detection of bacteria, and as such paved the way towards a theragnostic process where we can capture and identify microbes, for example, in sepsis patients. And I'd also like to highlight that capturing efficiency and degradation of the magnetic beads still remains a major issue in the translation of this process. Thank you.
For an interesting approach.
I think interesting and clinical, very relevant topic. 2 questions. First of all, you mentioned that you can decrease the pathogenic load after application of the nanoparticles, and you show this graph. How do you measure the concentration of the pathogenic load? Because you mentioned that there's a decrease. How mention you this? First of all, the second one, since we all know that sepsis is a very heterogeneous disease, you do have many sizes of components in the blood, bacteria or cell detritus, whatever. How do you can focus that you really catch most of them because they are so heterogeneous in size?
Excellent questions. So the first one, how we measured the pathogen load, we counted the colony-forming units. So that's a very basic way. Yeah, of course, there are more advanced ways to do that, but we'll definitely do that in future. The second thing is also a very good question. I think also in terms of endotoxins, that's also another really interesting application. Yeah, so it will really show in a clinical study whether removal of a bacteria is an important thing or whether endotoxin removal is more important or both. I think this mostly remains unclear and needs to be investigated, what will actually improve the patient's condition in the end.
You are now discussing bacteria and the toxin. Can you comment on other applications, maybe like autoimmune disease you mentioned in your abstract?
Yeah, I mean, we've looked at the different potential applications, and right now the bacteria or endotoxins are something that is relatively easy to investigate and to measure, and they are also in vivo models available to a certain extent. So that's why we started with that. But definitely, I mean, there are other really relevant applications that are worth to be investigated.
But that still needs to be started. You are not doing that in parallel already?
No.
Okay, no. Thank you. If not, no other questions, we proceed to the third speaker from here, Basel, Novartis.